Welcome. I'm Leah Schutz, the co-founder and president of the Families SCN2A Foundation. We are so happy to host Dr. Sanders and Bender at our webinar today. Dr. Kevin Bender is trained as a neuropsychologist and is an assistant professor in the Department of Neurology at the University of California, San Francisco. He holds an endowed chair in neurology in honor of the Gallo family and is a member of the Center for Integrative Science, Kavli Institute for Fundamental Neuroscience, and Will Institute for Neurosciences. Dr. Stephen Sanders trained as a pediatrician, geneticist, and bioinformatician. He's an assistant professor in the Department of Psychiatry, also at the University of California, San Francisco, and the director of the Psychiatry Department Bioinformatics Core. In addition, he is a member of the Will Institute for Neurosciences and the Institute of Human Genetics.
Thank you very much for that kind introduction. It is an honour to have this chance to talk to these to families about this work which we've been doing. And what we'd like to do today is to go through the important genetics and physiology work which we've done, which we think gives some early insight into understanding the way that SCN2A leads to the problems which we observe. I'd like to start just by introducing our two different voices. I'm Stephen Sanders. I have a slightly distinctive voice, and I'm joined also with...
Hi, I'm Kevin Bender. I have a less distinctive voice, I guess.
There we go, and we hope that will be clear, but if ever you want to ask which one of us is talking, just please feel free. So we're going to start off thinking with a little bit of background in genetics, because I need to take you through some of this biology so we can understand the genetics of SCN2A in particular. Some of this will be essentially a revision of things you may have learned at high school. Some of it may be a bit newer. So if I was to look inside our cells, inside our bodies, we are made up of trillions of cells. And here, you should be able to see a little red pointer. Here is an example of a cell. Inside almost every single cell, we find something called the nucleus, and that contains these little structures here, which are called chromosomes. So we can then zoom in on a chromosome, and this chromosome is made up of tightly coiled, essentially proteins and DNA. And you can see here an example of this DNA being uncoiled, zoomed in, and zoomed in, and eventually we get down to this single strand here of DNA. And if we zoom in far enough, we get to the stage where we can see individual nucleotides, and this is the A, T, C, and G, which people are usually familiar with in DNA. This DNA contains all the information you need to create a fully functioning human being. It is, along with being an information store, it also is key to how each cell functions. When I was at school, I was taught that DNA was like a book. Now working as a geneticist, I'd say it's much easier to think of this essentially as a little mini computer at the heart of every single cell. It contains the processing, the information, which allows cells to act as they're meant to. So if we then zoom out a bit, if I look inside one of these nuclei, I will find 46 different chromosomes. These come in pairs, so up on the top left, here is chromosome 1. They're numbered based on their size, so chromosome 1 is the biggest, 22 is the smallest. Each chromosome we have two copies of. One copy comes from your mother, one copy comes from your father. And then down at this far end here we have X and Y. So this would be a male. If it was female, there'd be two X chromosomes. These chromosomes are made up of DNA. In fact, each chromosome is one DNA molecule just coiled up very tightly. And we stain them with a special substance which makes bits of them appear white and other bits appear black and most of them appear grey. And it's this banding of black and white which allows us to identify which chromosome is which. Here is chromosome 2. So it's the second biggest chromosome, and that should now be marked and being ringed. If I was to zoom in on chromosome 2 and flip it round and stretch it out, then you can see these bands becoming much clearer. Using very high resolution cameras and microscopes, you can identify these bands and work out exactly where they lie. And so this is now a little schematic of where those bands lie on the chromosome. So here we're seeing two copies of the chromosome. Again, one is from the mother, one is from the father. We can't tell which is which just by looking at it. And this whole structure here is chromosome 2. Within it, we find this region here, which is called 2q24.3. The 2 is because it's chromosome 2. The Q means it's on the longer arm of it. And the 24.3 refers to exactly which band this is present in. If I was to then zoom in further, I would get down to the level of being able to see individual genes. So we've zoomed in massively here. The entire chromosome is about 2 million base pairs long. We're now looking at a region of about 2 million base pairs. Every single base pair in that chromosome is labelled with a number. So, for example, here's number 165,500,000. And this is from the Human Genome Project back in 2001, which essentially gives us these numbers. This distance here represents 1 million base pairs, and you can see that there is a cluster of genes here, many of which are called SCN. So we've got SCN 2A, 3A, 1A, 7A, and 9A. And the reason for that being simply that all of these sodium channels are quite similar, and during evolution they split, and that's why they all live as this little cluster here. But today we're interested in this one here, SCN2A. So if I zoom in even further, so we're now down to looking at about 50,000 base pairs in that scale there, we can now see the structure of SCN2A. It starts off over here, it is then read from the left to the right, and it ends over here. Now, SCN2A is a gene which is found in the DNA. One of the key things which DNA does is to contain information. And in DNA, that information is encoded by the A, T, C, and G, which I've shown here as the red lines on the DNA structure. For that information to actually do anything, it needs to be read and made into a protein. So for a gene in the DNA, basically the whole point of that gene is to make a protein. The way that works is the gene is read by this protein here called Pol2, which allows RNA to come off it. So here we have the double-stranded DNA, it is split apart, and the single-stranded RNA, which now contains the information, comes out of it. That information leaves the nucleus in the centre of the cell and goes to this structure here called a ribosome, where the RNA at the bottom is then translated into a protein. And that protein is, in this case, the NaV1.2 sodium channel. So from the point of view of SCN2A, the gene in the DNA becomes the SCN2A RNA, which becomes the NaV1.2 channel. This is zooming in again on a gene. This is just an example of a gene. SCN2A looks very much like this. Over here we have the promoter region. This is where the Pol2 enzyme binds. It then reads the RNA all the way along here until it gets to the end, and then that is what then goes out to make the protein in the nucleus. In terms of which bits actually make the protein, the majority of this gene doesn't. These regions here called introns get removed and essentially don't make a protein. The bits which do make the protein are the exons, marked here from 1 to exon 6. So the way that DNA makes a protein is essentially every 3 base pairs of the DNA, so for example CGG, encodes 1 amino acid. And so the protein is made up of amino acids in the same way that DNA is made up of base pairs. So if I was to look at the SCN2A gene, there's about 6,000 base pairs, which makes about 2,000 amino acids. This code here, CGG, encodes the amino acid arginine, which we represent with an R. That's because there's another one beginning with A. If we change that to a CAG, then that encodes a different amino acid, this time glutamine, which we encode with a Q. There's lots of other ones beginning with G. And so you can see here that a single change in one nucleotide from a G to an A makes the amino acid change from an R to a Q. And this is a concept which we call a missense change. So I want to take you through now an example of receiving a genetic test report. And the data here, the information here, should be comparable to that which you would get for a genetic mutation in SCN2A. I'm going to go through this for a missense variant. At the very end of this talk, we're not going to show them today, but I've included the same details for other types of variants which you can have. And at the end of this, we're going to make these slides available, and you should find that whatever mutation was found in your child, you should be able to translate that into one of these types of mutation. So I said that every single base pair was labelled. Let's look at this base pair here, which is on chromosome 2, position 166,198,975. A G changes to an A. So there's the chromosome, there's the location, and there's the expected and changed nucleotide. So this is what changes in the DNA. If I was to look in the RNA, which comes off this, the RNA essentially has 6,018 bases in it, and this position here represents position 2,558 in the RNA. And we see the same change, a G to an A. If I was to then look in the protein is a third the size of the RNA, so it's now 2,005 amino acids, and this base pair here results in a change in the amino acid at position 853 from an arginine to a glutamine. So this takes you through the three different ways of representing the same thing. I can describe it in the DNA, in the RNA, or in the protein, but all of them are saying the same information. So we've described here, this is a simple example of this. We have two copies of the gene SCN2A, one from the mother, one from the father. Both of those get changed into RNA, which leads into a protein from the mother and a protein from the father. Over here I've shown that example of a missense variant. I've shown it being present in the father, it could equally be present in the mother, and so that change in the DNA in the father leads to a change in the RNA, which comes off the father's gene, which means that one out of the 2,000 amino acids in the protein is changed in the father, but the one from the mother still remains the same. So essentially, 50% of the proteins from SCN2A now have this one little change in them. Now that accounts for probably the majority of mutations which we see in SCN2A, but you'll find that, I expect that individuals in this call will have another type where instead of causing a missense mutation, you have a truncating mutation which stops that copy of the protein being made. Let me take you through the logic of that again. We start here with GGA in the DNA, which encodes the amino acid glycine, which is shown as a G. If that changes from a G to a T, we get TGA in the DNA, and this time that encodes a stop codon. That's how the gene, or the ribosome, knows when to stop making the protein. If that happens too early, then you're going to get a protein which is much, much shorter than it should be. And in fact, there's special machinery inside every single cell which stops that from happening. Instead of getting a shortened protein, some special machinery comes along and chews up that RNA, so in fact you get no protein being created. So going back to my simpler schematic, over here we have a gene from the mother, we have a gene from the father, there's a single base pair change there, you get the RNA, but one copy of that RNA gets chewed up, so you only get the protein coming from the mother, but you might expect to have half as much of that protein because the father's one won't be present. And again, this could equally happen from the mother or the father. There are several different phrases used to describe this. Sometimes it's called a loss of function, or a protein truncating variant, or likely gene disrupting. It can also be called nonsense, or frame shift, or splice site mutations. All of them are the same. They result in this absence of that one copy of the protein. And before we move on to the sodium channel itself, there's just one more bit of biology we need to think about, and that's how that genetic variant arrives as in the child. The vast majority of the DNA changes which you have are inherited. So we have about 3 billion base pairs in our DNA. In every single one of us, about 3.5 million of those are variants, which are what make us unique. and the vast majority of those are inherited. I'm showing here an example of a rare inherited variant. This is rare in the population, not many people have it, but it's passed on here from a mother to a child. And an example of this, which you may well have seen in day-to-day life, is piebaldism. So this causes a little flash of white hair and underlying lack of skin pigmentation, and this tends to run in families, getting passed on from one parent to their child. On the right side, I'm showing the idea of a de novo mutation. This was not present in the parents, it's not present in the fathers or in the mothers. It is a new mutation, and that's what de novo means. It means a newly arising mutation. These are extremely rare. Out of those 3 billion base pairs, in each of us there's about 70 which are new coming from our parents. Because these have not been essentially tested by natural selection, these have the potential to carry a lot of risk. An example here would be Down syndrome, where a de novo mutation can cause essentially the entire constellation of Down syndrome, and that generally can't be inherited because people with Down syndrome are unable to pass on their DNA. So let's move on to the channel itself. So here's an example of the NAV1.2 channel, which is created from the SCN2A gene. It sits here at the surface of a cell, and it essentially forms a hole which can open to allow sodium, shown here as the little blue dots, to pass into the cell. It's called a voltage-gated sodium channel because it opens in responses to changes in voltage, essentially to an electrical signal. And throughout this, we're going to refer to sodium as Na+, which is its chemical symbol of the ion. So to understand how this channel works, the key thing is it responds to voltage and it opens up to allow sodium through. So this is another way of showing the resulting protein, the NAV1.2 channel. If you remember, I said it was made up from amino acids from the mRNA. Those amino acids are created in a line. Starts over here, and every amino acid is added until you get all the way over here to number 2000. You can see it's essentially created in one long line. But it needs to make a channel, and so the way that happens is you have these 4 repeating units. So labelled here as 1, 2, 3, and 4. So there they are. There's 1, there's 2, and there's 3, and there's 4. Those essentially fold to make a circle. So we've shown here this example here. There's 1, there's 2, there's 3, and there's 4, and they folded up to make this channel. In the centre of it is this hole here, and that's where the sodium, shown by Na, passes through. And for that to happen, you need these 4 very special amino acids. They're labelled here in black as the ion selectivity filter, and here they are here. I'm going to mark them with a little red pointer. There's 1, there's 2, there's 3, and there's 4. And so those are held very closely together to make essentially a channel through which only sodium can pass. It won't allow through potassium or calcium or any other ion. Only sodium is allowed to come through. And it is critical for this to work for those 4 amino acids to be held in the right location. We've also marked here what's called the transmembrane domain 4. You can see there's 4 of them. And it's marked in blue with these pluses on it because this is where the change in voltage is sensed. And if we knew nothing else about this channel, we might expect the bits which are the most important would be this voltage sensor and this section here which holds that ion selectivity filter to make it allow sodium to come through. So it's been known that mutations in SCN2A can lead to disorders for about probably 20 years or so now. The first ones discovered were the associations with epilepsy. We only really became aware of its function in autism and developmental delay back in 2012, which was some work done by the lab I was in at the time using some new technology to look into autism spectrum disorder. So we have this slight mystery here that this gene, SCN2A, can lead to 3 different disorders. I just want to go through each of those 3 I hope these descriptions are enough that most people could recognise which one of those they might fall into. The first symptom we see is called benign infantile familial seizures, or BIFs. It's sometimes used with another acronym here, which is benign familial neonatal and infantile seizures. They're really the same thing. This is basically characterised by the onset of seizures before 12 months of age. However, these seizures generally resolve, often by 1 year, but almost always by 2 years of age. And amazingly, these seizures resolve without any long-term developmental or neurological issues. By the age of 10, you would not be able to recognise these people on the street from someone who's had more severe seizures. From someone else walking around. There's another symptom which is characterised by early onset seizures, and that's epileptic encephalopathy. This is seizure onset before the age of 12 months, often seen at birth, often reported before birth in utero. These seizures generally continue beyond the age of 2 years. Sometimes they seem to resolve after many years, sometimes not. But critically, they're associated with developmental delay, which is often severe. And then the third symptom group we see is autism spectrum disorder and or developmental delay. These individuals are characterised by not having seizures before the age of 12 months, but they do have delayed developmental milestones. This might be walking slow, this might be talking slower than other children. And once they get old enough, they can have a diagnosis made of autism spectrum disorder, often which is on the low functioning or severe end of the spectrum. Some other children don't have a diagnosis of autism, but do have often global developmental delay. And when a child gets old enough, that global developmental delay may also be called intellectual disability. After the age of 12 months, a small number of these individuals may develop seizures as well. It's unclear to us exactly why that happens, but this is a common finding in individuals with autism or developmental delay, that they often get late onset seizures. It happens in about 20% of people with autism. And so our mystery as scientists and as geneticists is to understand how can this one gene create these 3 different disorders. And to explore this, I'm having a little problem here, the slide's not advancing. To explore this, we started to look at how those genetic variants lay on that protein. I'm having a little technical issue here. Oh, there we go. Fantastic. Good. So what we did was to basically read through the entirety of the literature on the gene SCN2A. We went through and looked at over 148 different publications, each of which mentioned a genetic variant within the gene SCN2A and associated with some symptoms. From this, we found about 140 different genetic variants, and here I'm showing the ones which are associated with this benign infantile familial seizures. All of these variants were missense. These are the ones which change just one amino acid out of the protein. In addition to that, many of them were inherited, so that's running in families, though a small number of them were de novo. The fact they were inherited fits with this having a milder phenotype. When we looked at these, we saw that they might cluster around this voltage sensor, this transmembrane domain number 4 in these 4 areas. We then looked at the ones associated with epileptic encephalopathy, so that's seizures before the age of 12 months, but long-term developmental delay. Again, all of these were missense. That means they affected one amino acid out of the 2,000 in the protein. Almost all of them were de novo. That means they were not inherited, and that fits with the more severe symptoms. Again, we saw them clustering around this transmembrane domain here. We then repeated that, looking at the variants which are found in autism spectrum disorder and developmental delay. This time we saw 2 different sets of variants. The first set were these truncating mutations. All of them were de novo, and all of them resulted in one copy of the protein not being made. you can see that these are shown throughout the gene. It doesn't really matter where these mutations are, because they result in a stop codon, which means that the RNA is chewed up, so you don't get the protein. It doesn't really matter where they lie. What is important, though, is that all of the ones which destroyed this one copy of the protein were found in autism spectrum disorder and intellectual disability, not in epileptic encephalopathy. And then the last set of variants we saw was, again, the missense mutations. So that's missense being only one amino acid out of the 2000, de novo being newly inherited from the parent, fitting with the severity. Now, with these missense mutations, the location does matter. And what we saw is that they generally do not overlap with the ones we saw in epileptic encephalopathy. Instead, they seem to cluster in this other area, which we think is important, near the pore, which is allowing this channel to allow sodium through. So this led us to the idea that the mutations in autism spectrum disorder and developmental delay were acting in a different way from those causing seizures before the age of 12 months. And in fact, because this included, on the autism side, ones which were disrupting one copy of the protein, this led us to the theory that these missense mutations in autism were resulting in the protein not working, whereas the mutations seen in infantile seizure were resulting in the protein essentially having a gain of function, working too well and letting too much sodium through. At this stage, we didn't have any evidence to back that up beyond the location of these mutations, but it created to us a very compelling hypothesis. And as we look at more data coming in and add in more autism mutations, we found that this idea seemed to be replicated, with them hitting the same locations again in this pore loop. So at this stage, we have this idea that the epileptic encephalopathy mutations are causing a gain of function with too much sodium being transmitted, whereas the autism mutations are causing a loss of function with not enough sodium being transmitted. And at this stage, I turned to a dear colleague at UCSF who works on electrophysiology, which is the study of how sodium goes through a channel like this. And in the same way that I passed this scientific question to him, I'm now going to pass this presentation over to him.
All right, great. Thank you very much, Stefan. This is Kevin Bender again. And I wanted to run you through what we did based on what Stefan was telling us in terms of the genetics to try and understand how the electrical activity that is mediated by SCN2A or NAV1.2 is altered when we start to examine individual mutations that are found in individual children. So for this, we need to go through a little bit of background, just like Stefan did in terms of the genetics, a little bit of background in terms of now you have to turn into an engineer and understand electrical activity occurring within brain cells. And so the first thing we should talk about is this sodium channel, SCN2A. As Stefan mentioned, when the channel opens, it allows sodium in. Now, one interesting thing about sodium is it carries a charge. It carries a positive charge. And so what happens here is you go through this insulation area, this little membrane here. It's actually a perfect insulator between the outside and inside of the cell. But when you have a positive charge going from the outside to the inside, it does something called depolarize the neuron. And depolarization is a way of increasing the voltage within the cell. It's the same thing as sort of flicking on a light switch and turning on a light. It's essentially turning on a light very quickly within a neuron. And those flickers of light are called action potentials. They're sometimes called spikes. And so this is a unit of information that is used by brain cells to transmit information from one cell to the next. And the whole combination of information passing from one cell to the next is what gives rise to our ability to speak, our ability to move, our ability to experience the world. So at the basic level, an action potential is mediated by two different things. At the very start, you get a lot of synaptic activity that leads to small depolarizations of the cell. Eventually, right around here, you get to what's called a threshold, at which you can activate a sodium channel. And remember, it's voltage dependent, and so it cares about a specific voltage at which the channel will open. When that occurs, sodium enters the cell, and you can see that the voltage inside the cell increases, and it depolarizes. Eventually, potassium channels, which are very similar to sodium channels, except for one key thing, that is they allow potassium to flow through. Potassium channels open. And what's interesting about that is that potassium, instead of coming into the cell, potassium, which is also positively charged, goes out of the cell. And this allows a neuron to repolarize. And that gives rise to this very characteristic wave of activity called an active potential or a spike. Now, as Stefan mentioned, we have a condition here where SCN can give rise to either epilepsy or autism spectrum disorder, and we have this hypothesis that the way in which individual mutations were affecting the channel could give rise to either gain of function or loss of function in the channel. Now, what does that mean for an actual neuron? Well, one way of thinking about it is in terms of the electrical activity. And so under normal conditions, you can have a series of action potentials generated by an individual cell. And these will go on and they vary in time, they vary in salience. If you have a condition where you have a gain of function, where either more sodium is passing through the channel or the SCN2A channel responds differently in response to voltage, it's a little easier to open for a given voltage, you can have something called a gain of function. And I hope you can appreciate here that there are just more spikes, more action potentials in this train than in a normal train. And this excess activity gives rise to epileptic activity within whole networks of brain cells. The converse of that is a loss of function mutation, where it's either harder for a channel to transmit a given amount of sodium, or the channel responds less well to a given voltage. And in this case, the neuron fires fewer action potentials in response to a given stimulus. And this is what we would equate with a loss of function at the neuronal level. So our real task here is to understand whether gain of function or loss of function occurs with these various missense mutations that we identified previously. And this is how we have to step through. All of these missense mutations that we've identified previously in SCN2A. So these are new mutations that we've identified recently. We want to understand how they map onto the electrical activity of the cell. So to begin first, we have to actually take a step back from brain cells and go into a different regime of electrical recording. And this is something called voltage clamp recording. Now, I won't really explain what that really means, except for this is a way of doing very careful characterization of the electrical activity of the SCN2A channel, the NAV1.2 channel, in isolation. So instead of studying brain cells at this stage, because brain cells have sodium channels and potassium channels and other voltage-dependent channels, we instead did this in what's called an HEK293 cell. And what's important about an HEK cell, it's actually a human embryonic kidney cell, is that they do not have any sodium channels or potassium channels themselves. And so you can do fancy tricks with modern genetics and modern molecular techniques where you can introduce a channel of your choosing and of your design into these cells, and then the cells make more of these channels and they insert them into their own membranes. And once you have that condition, you can then study the electrophysiological characteristics, the electrical properties of these channels in isolation. And so here on this old TV screen, which we have in lab, you can see the shape of an HEK cell. It looks a little like a crater on the moon, but this cell in isolation is chock full of NAV1.2 channels. And you can see this very blurry line right here. That's actually a glass electrode filled with a special salt solution, and we use that solution to make a recording directly from the inside of these cells. And when we do that, we can then introduce various voltage steps. We can basically change the dimmer switch on the light in which we're activating the cells with electricity. And the cell responds in a certain way. And this response are these downward deflections in current here. And for some reason that we don't really need to go through today, the upward deflections that you saw here in the action potential are equivalent to downward deflections in voltage clamp. And so this is the cell responding, the sodium channels responding more and more effectively to these increases in voltage with more and more current as sodium enters the cell. And so that's our general technique. And with that, we can start to ask questions about how the physiology of these channels has changed in response to various mutations. And the first thing we started with is we wanted to verify that the system was working in our hands. And so we started with what are called wild type or normal channels. And you can see we got a range of responses in the amount of current that we got, and that's plotted down here, normalized to the size of the cell, because different cells have different size. The more membrane you have, the larger the current you get. But on average, we got about 500 nanoamps per picofarad. Importantly, when we record from cells that were not transfected or in which we did not insert our new channel, we did not see any current. That suggests that the system is working well in our hands. The other important thing that we checked is that there's a prediction that what are called protein truncating, loss of function, or nonsense mutations, splice site mutations, should give us a condition in which no channel is formed and therefore no current, no electrical activity should be observed. And so here's one example of that. This is called C959X. And you can see we try to activate the channel, but there's basically no current except for a little bit of noise generated in this case. And we checked that for a few different variants that have been identified. And the story was the same in all of these, that there was no current generated. We next turned to asking the question about the missense mutations. And when we started to look at this, we started to identify various effects. One of the most striking effects was resulting from those mutations that are found at the pore that Stefan mentioned. Some examples of that are R379H. This is a mutation that's just a few amino acids away from the ion selectivity filter in the third repeat of the channel. And in this case, what was remarkable to us, what really surprised us, was that, again, just like a protein truncating variant, we saw no current. And it turned out that this was the same for other mutations that occurred at this exact same amino acid, and a similar mutation found earlier in the channel. One other thing that we did is checked to see whether or not the channel changed its ion selectivity. As Stefan mentioned earlier, these channels are remarkably selective for sodium. Sometimes when you get mutations like these, you can change the selectivity of the channel. You can turn it into a potassium channel, or a channel that doesn't really care as long as the ion is positively charged. That did not appear to be the case here, because when we did various techniques within our lab to test for those things, we again saw no current passing through the channel. Okay, the next question we wanted to ask was whether when we express these channels in a condition where that would replicate what happens within the child, where you have one normal gene and one mutated gene, you get what you would expect. And so that's shown here with a co-expression of wild type and one of these channels that gave us no current. And we predicted that we would see something around the middle, something around the average between these two, and that's indeed what we saw. But then we checked 3 other mutations, and these became a little more interesting, a little harder to interpret. Here we looked at 2 mutations that are found on the N-terminus, and those are D12N and D82G. And you can see that D12N was, from this view, looked normal, whereas D82G looked to be a channel that produced less current flow. The same was true for T1420N. Again, that surprised us because this is actually quite near the ion selectivity filter. We were expecting this to be similar to its brethren over here, but in fact, it provided a good amount of current. And so now the question becomes, well, how do these mutations, which give rise to channels that are somewhat functional, mirror the effects that we see in cases where we have a channel that is completely non-functional. And to do that, we had to do a little bit more of an electrical assay, and that was to measure various aspects of the channel's function. And so as I mentioned here, this is a case where we're activating the channel, and we step it through various voltages, again activating, turning on the dimmer switch from very dim to extremely bright. And you can see that as we step up and up and up and up and up, we get more and more flux of sodium through the channel. The opposite is also important to study with these sort of mutations. And this is a case where we hold the channel at a different voltage and then we step it to a case where it should be fully activated. Well, its history, the voltage history before it got fully activated, determines how many channels are available, because the channels go through something called inactivation. Essentially, a channel once opened has to then repolarize down to a very low voltage to allow it to then enter what's called a closed state to allow it to then reopen. And so it's a three-step process. You don't just go on and off like a dimmer switch. You can imagine you have to turn your dimmer switch all the way on, and then you can't do anything else except to shut it fully off. Otherwise, it will not go back to a new state, a new level of light. And so that's what inactivation is. And you can see that if at very, very hyperpolarized or very low voltages, you have full availability of the channel, but it goes down as you depolarize the cell. You increase the voltage. These are normally shown overlaid when you look at papers that are reported in this sort of with this sort of characterization. And so the inactivation and the activation curves are shown overlaid. And one thing that's important to realize about this is that shifts in these activation or inactivation curves can tell us quite a bit about how individual mutations affect channel function, and can tell us a lot about gain of function or loss of function or modulation of function in various cohorts. The other thing that could happen within these channels is it could activate or inactivate at the same voltages, but it could do so more quickly or more slowly. And so the channel has something called a kinetics. It's basically the speed at which it opens and closes. And you can see that this example, it's very slow and it increases in speed. That whole ensemble of speeds could change depending on individual mutations. And that's another thing that we examined. And so here's an example of one of those mutations or variants that we've identified as still producing a level of activity, but an altered level of activity. And we identified why, and that's in the case of D82G. Here, there's no change in the way the channel inactivates or the amount of the number of channels available at a given voltage. But you can see that compared to what's the wild type or the normal case, D82G was harder to turn on, harder to open in response to a given voltage. And so to get half of the channels activated, instead of needing something like -30, you need something like -20. So it's a 10 millivolt shift, give or take, in the level of activation, and that's plotted over here. The other thing that's plotted over here is all of the other variants that we looked at, as well as this half and half case. And what's important to show here is that we basically did not see any other changes statistically. But we looked at a few other aspects of these other channels, and we did find, I'm not going to show it today, but we did find that D12N closed more quickly. D82G, as we're showing above, was less responsive to voltage, and T1420M, which is very similar to D12N, also closed more quickly. And so now we have a situation where we have various effects. We either have a channel that has no function, or as a channel that is destroyed at the level of mRNA, or a channel in which it responds differently within the cell. Now, one thing I should mention is we're just talking about a few mutations here today, but this is just a few of a larger cohort that have been studied in more detail. And if you refer to this table, which will also be linked below, you can identify information that has either been done by ourselves or by others in the field in which other mutations or other variants have been characterized. Now, that's going to be a relatively small subset of the total number of variants that have been observed. And this is one thing that many in the field are working on, to really understand in full what happens with various mutations associated with BIFs, EE, and autism spectrum disorder. But for now, what we'd like to do is show you how we take all of this information we get at the level of voltage clamp in these HEK293 cells and start to develop a working model of how they would affect neurons. Once we incorporate these various results into neurons, what effects might occur. And so for that, we need to first start by understanding where SCN2A or NAV1.2 is expressed in the brain. And so this is an example from a mouse. This is not a human. And this is a cross-section of a mouse brain. This is one of the tools that we and others use in lab as a way of understanding neuronal function in model organisms, which we then can relate to what goes on in humans. And three important areas light up here. One is the cortex. This is basically the outer shell of your brain. Underneath this is the hippocampus. This is where a lot of spatial memory and spatial recognition and short-term memory is encoded in your brain. And also the cerebellum. You can see it lights up quite heavily in this stain for NAV1.2. The cerebellum is involved in motor function and error correction. So we decided to focus first and foremost on the function of NAV1.2 in the frontal cortex, in the part of the brain that's associated with cognition, with memory, and with social interaction. And so within this part of the brain, NAV1.2 is primarily expressed in cells called pyramidal cells, which are excitatory cells. They turn on other cells when they fire an action potential. And within these cells, NAV1.2, or SCN2A, undergoes a very interesting developmental progression. And so early in life, SCN2A is the only type of sodium channel that is found within these cells. And it's found in a part of the cell called the axon initial segment, and this is where neurons transform inputs into action potential outputs. And so you could imagine, since it's the only channel involved in action potential initiation at this stage, it could play in a very important role. And mutations or variants in this channel could have some very interesting effects on neuron function. Later in life, more sodium channels start to become expressed in pyramidal cells. One important one is called NAV1.6. It's encoded by SCN8A. You can see that it starts to occupy a large fraction of the axon initial segment. NAV1.2 is still there. It starts to occupy now the proximal part of the initial segment. But if we go back and we look at the voltage-dependent characteristics of these channels, this is the endogenous or native voltage-dependent characteristics of these channels, you can see that their lines are shifted relative to each other, such that NAV1.6 is always on the left side. The fact that it's on the left side is important because that means that NAV1.6 responds more easily to a given voltage. And so when a cell is depolarizing in response to synaptic input, NAV1.6 channels will activate first. And so in this regime, when we have a mature cell, action potentials will initiate in this NAV1.6 rich regime and then propagate in two directions. Okay? So this sort of switch can actually provide us with some very important insight into what might be going on in terms of benign infantile seizure and epileptic encephalopathy. And so if we model the voltage-dependent characteristics of the channel as a function of development, early in development we only have to worry about two curves, the curves of NAV1.2. But in a mature, normal cell, we have to worry about four. Here we have NAV1.2 and also 1.6, where we have these subtle shifts in voltage dependence. Now, in the case of benign infantile seizure, one mutation that has been characterized quite well in this paper down here is the case of L1563V. And in this case, what happens is the wild-type activation curve, the way in which the cell responds to which the channel responds to voltage, gets shifted a little bit to the left. That makes it a little more easily excitable. Now, early in life, you would predict that that could make the entire neuron more excitable. But what's interesting is later in life, when you get expression of NAV1.6, you can see that the mutated curve overlaps quite well with the wild type or the normal curve of NAV1.6. And so in this case, you would actually predict that the excitability of the neuron might return to normal. And that might explain the resolution of seizures that we see somewhere between 1 to 3 years in life. That's in contrast to what you see in EE, or epileptic encephalopathy. One mutation that was identified and characterized there is I1473M. Here, there's a rather remarkable shift in the activation curve of NAV1.2. And it's so far left shifted that that this channel remains ever more excitable than even the wild type NAV1.6. And in this case, we might have hyperexcitability. So now what's nice is that we in the lab can model these sorts of changes in excitability in terms of computer models, where we can take what we know about a neuron's shape, a neuron's size, as well as the distribution of all of these different channels. And as I mentioned, we know where NAV1.2 is in a developing cell, and we know where NAV1.2 and NAV1.6 are in a mature cell. We can take all of that information and then ask how these different mutations would affect the activity of individual neurons. And so that's one thing that we looked at. One prediction from the previous page is that epileptic encephalopathy mutations should give what's called hyperexcitability or gain of function throughout life. And so that's what we looked at first. Here is a spike train or an action potential train from a wild type cell. And then what we did is we modeled a heterozygote condition where we have one normal SCN2A gene and one SCN2A variant found in EE. And in this case, we did I1473M as well as another variant E1211K. And in both cases, we saw far more action potentials generated than the wild type condition. When we looked in a mature model, we still saw that there were more action potentials generated, not as severe as what we see in early development, but certainly far different than what you would expect from a wild type cell. In contrast to this, when you look at benign infantile seizure, you find that the cells are hyperexcitable, or they fire more action potentials in early life in a developing model where only NAV1.2 is expressed, and that's shown here. But then when you get into a mature model, you get this largely resolution in excitability, where excitability pretty much comes back to normal. And this is consistent with what we were seeing in the clinic. Okay, so now our big question was to ask what is going on in autism spectrum disorder. Here we have a variety of effects. We either have these small changes in voltage dependence of activation in the case of D82G. This is just showing the shift in kinetics, the shift in the speed of the channel in D12N and D1420N. And we took all of these different parameters and we incorporated it into a model of action potential initiation in these cells. And what we found is, in contrast to what we saw with EE and BIFs, we saw less excitability, essentially a loss of function. So I want to run you through the important parts here. So first, there's the wild type cell in the developing case where it's only NAV1.2. Then we modeled the case where we had a protein truncating variant. So here we have one wild type or one normal SCN2A gene, and then we have a case where no SCN2A gene, or sorry, no NAV1.2 protein is made from the other allele, the other gene. Now this will be equivalent to many of the cases of missense variation, for example R379H or R973H, where it's not a loss of the gene or a loss of the channel, but the channel does not have any function at the membrane. And so this models both loss of function and many of the missense mutations that we call non-conducting. And you can see that far fewer action potentials are generated in this case. The same is true for the other variants. And so all of them were shifted in the opposite direction as what we saw with the epilepsy mutations. But interestingly, just like benign infantile seizure, we saw a resolution for the most part when we incorporated it into a mature pyramidal cell. And so this would suggest that early development is a potential critical period for excitability. That's not to say that other things might be going on in a mature pyramidal cell that could lead to continued alterations in neuronal function. And that's something that we're interested in exploring in the future. But overall, it leads us to this model of understanding de novo mutations and inherited mutations in SCN2A, where you have a normal regime and those mutations that make the channel less excitable could lead to autism spectrum disorder and developmental delay. If you have minor gain-of-function mutations, then you can have something called benign infantile seizure. More severe gain-of-function mutations can lead to epileptic encephalopathy. At this point, I want to turn back over to Stefan because he has some interesting findings in terms of other phenotypes associated. Yeah. So we'll turn back to that and we'll wrap up within the next few minutes.
So Kevin's shown here our model, essentially going from the autism side with loss of function to varying degrees of gain of function, which is distinguishing between benign infantile seizures and epileptic encephalopathy. This is a model. It is not the complete answer to everything. It is just meant to be a way of us starting to think about the biology. So far, what we've seen of new data coming in, it generally seems to support this model as being a useful framework, but we do expect to see some exceptions, and those exceptions will probably show us the most about understanding the next phase. But we are very, very clear that this is not explaining all of the symptoms and signs which people are seeing. So let's start off with, I think, what to me is the biggest mystery. It looks like many people are presenting with ataxia. This is clumsiness, stumbling when walking. It can be associated with slurred speech as well. It seems that those symptoms are somewhat episodic, as in they appear to come and go. I originally thought that those were just associated probably between epileptic encephalopathy and benign infantile seizures. But that seems to me now that actually it seems to happen on the autism side as well. At this stage, I simply do not know how these two different groups relate to the ataxia. We know that this is expressed in the cerebellum, where ataxia tends to be common, and I think this is one of the big questions where scientists need to work with the patients to try and work out what's going on there. Focusing on the autism side, we know that some people have late onset seizures. This is not that surprising in autism spectrum disorder. Whether it is more common in SCN2A, I think is difficult to know, but that's, again, something we're interested in finding out. And then there's been a couple of reports suggesting there's an association with schizophrenia. It doesn't look like people with autism or developmental delay move into schizophrenia. It looks like some people have such mild symptoms that they present primarily with the schizophrenia later in life. On the other side here, we see these symptoms which are associated with it. It seems very common to report hypertonia, which essentially is having sort of floppy, maybe weak limbs. There's also been reports of dystonia, which is essentially difficulty with movement or very tense movements. And what seems very clear is there's an association with microcephaly, that's having a small head, and cerebral atrophy. And so that's essentially the brain being smaller than expected. And I saw a question about polymicrogyria. While I'm not aware of certainly many cases having that particular diagnosis, I think there is an overlap between cerebral atrophy and polymicrogyria. Really the question is at what stage in development that happens. Those could well be part and parcel of the same thing. What this really captures, I think, though, is that we are just beginning this journey of understanding the relationship between SCN2A and the symptoms that people have. Understanding it is critical. Even this first model here changes the way we start thinking about the downstream experiments. We need to work very carefully with patients and try and really map this out. Essentially, we need to split things into two groups. Some things are related to the specific mutation. So, for example, we've seen in the mutation R853Q that it's associated with chorea, which is a very distinctive type of movement disorder. That to me looks like a true genetic association, and I expect some of these other features will be the same. There may actually be that in the future we can predict the degree of severity and even predict specific symptoms. Other symptoms will simply be down to what we call background risk. It is essentially what makes that individual unique, and that causes a range of symptoms in different people. So going back to our Down syndrome analogy earlier, in Down syndrome we see a very, very wide range of abilities coming with that. Some people live an independent life, most people do not. Some of that comes from essentially your background variation, what makes you unique. Some of it comes down to the severity of the mutation. And we need to work out exactly which one of those factors is taking place in SCN2A. One burning question we want to ask is to be able to work out who has a gain of function, who has a loss of function, and what degree of severity that is. We are early on in this, but we are starting to make some tools which might help with this. I want to just quickly show you one of the first ones we've created. I hope this is working, and you can now see, there we go, my web browser. This is an online tool which allows us to predict where in the gene a specific mutation occurs. We're going to make the link to this available in our slides, but if I just pick a mutation at random, well not at random, one which I know is going to work, R379H, we can see that it shows us that mutation occurs near the pore, and then when I hover over it, it tells me it's a missense mutation. This browser works for every conceivable single nucleotide variation. It's not yet working for frame shifts, but we'll add that in later. It also allows you to convert between these three different ways of representing it: the DNA, the RNA, and the protein. This is available to use right now. In the future, I hope this will also include information from the literature about what is known about each mutation. There we go. And then we've also been thinking about next steps. This is where we're stepping away from just our research, but thinking for the field. What do we need to achieve? We're going to start off with two things which are, well, one thing which is relatively simple and the information is there. We want to develop best practice treatment guidelines, both for the gain of function and also, if possible, for loss of function. This is really a question of collecting the information from parents, trying to work out what's worked, what hasn't, and then trying to make some guidelines for that. This would mean that when people are diagnosed in the future, they can go with those answers rather than having to find them out slowly. The next thing which we need to try and do is to predict the function of each mutation. At the moment, I'd say about 20 of them have been characterized out of the near enough 12,000 possible ones. We need to find ways to be able to predict how severe this mutation will be without having to laboriously go through and do the work which Kevin has shown us here. Going beyond that, our next big question is understanding the biology of how both the gain of function and loss of function variants lead to symptoms. This is just the first step. Recognizing that these two are different and that's leading to different symptoms, we then need to understand some very key questions. For example, what cell type is involved? Does its function change over development? And if so, what long-term changes can result from these? These are really critical to understanding how we're going to make progress in understanding how to treat these changes. And then that brings on to the last thing, and that is investigating therapeutic options. There are lots of groups working on this, as there are on the biology. Part of this is looking at simple drugs, which you might take by mouth. Another group is looking at proteins, which can be injected. And of course, being a genetic disorder, we're looking at ways of trying to think about gene therapy. But all of these therapeutic ones build on this foundation of understanding how SCN2A leads to changes. We're going to end here and now take some questions. I would just like to acknowledge the two labs which have been involved in this. On the left here we have Kevin Bender's lab, including Roy Ben Shalom, who is critical to doing a lot of the work here, and
that's Caroline, and she did a lot of work as well on this project with us.
Great. And over on this side, there's June, who helped me with some of the genetic sides. That's me, and Cindy created the web browser, which I just showed you. So we're now going to turn and answer some of the questions. I think probably the best way of doing this is if Leah or Angie coordinates.
Well, we've got two questions.
We've got two questions. All right.
Yeah.
So should we read the questions out first to make it clear which ones we're asking? I'll let you read out.
Sure. The first question is, how many people have been diagnosed with this mutation? And I think it means SCN2A as a group. And of the 3 types of mutations, which is most common and which is most rare?
So that's a good question. So to date, I would say that there's been, in the literature, there are about maybe now 210 people with SCN2A known variants. There are probably more people who've been diagnosed in clinics but haven't made it into the literature, and it's very, very hard to know what that number is. It's a number which is going to be going up very rapidly. Right now, we know of more mutations which lead to epileptic encephalopathy than to autism or benign infantile seizures. I think the reason for that is that the epileptic encephalopathy symptoms are so severe and so distinctive that many people who have those symptoms end up having genetic testing. Essentially, if you have a seizure in an infant, that leads to genetic testing, whereas if you have autism spectrum disorder in a 3-year-old, that leads to a referral to a psychiatrist and behavioural therapy without genetic testing. If I had to guess in terms of the total population affected, I would say that this is a relatively common disorder affecting autism spectrum disorder. I expect there to be more people affected on the autism spectrum loss of function side than there are on the gain of function. My logic for that being that it's very easy to break something, it's much harder to make it do something different, and generally loss of function is more common than gain of function. To give numbers to that, I would say that the loss of function mutations are likely to be about a similar frequency to Dravet syndrome, which is SCN1A, where there are now well over 1,000 people who are known, whereas the gain of function I expect is going to be probably in the hundreds in the same stage. For benign infantile seizures, that runs in families. That is rare enough that I've never seen a case. I expect that's probably the rarest of the 3 conditions.
Okay, the next question is a question about one of my slides. So as neurons mature, the action potentials no longer look like loss of function in the case of autism spectrum disorder. Does that mean that action potentials are now firing at the correct rate and no longer loss of function? Well, I would say that within that model, that is the prediction that we had, and that largely comes from the fact that NAV 1.6 has come online and allows and has basically taken over this job of initiating the action potentials. Now, one important thing to realize there, and this is a little too complicated to really discuss in that case, but you're very savvy in this question, is remember that NAV 1.2 got displaced, but it now starts to occupy this part of the cell called the proximal initial segment. And its role there is supposedly, or actually it's been shown with some modeling, in helping action potentials do something called backpropagation. So I talked about how action potentials are communication from one neuron to the next. There's actually also a reverse direction in which in which an action potential will flow, and it goes back into the dendrites of these cells. It goes back to the synapses. And at that point, it's been shown that action potentials are very important for a variety of processes in a cell, including synaptic plasticity, which is a form of learning within individual neurons, as well as encoding other interesting events, including activity-dependent gene transcription. And so all of those things, those are things that we didn't really capture very well in these initial models, but we're very interested in looking at in the future. And so it might be the case that while the action potential frequency or the number of action potentials has come back to the normal case, there may be an instance where there are continued alterations in the way neurons function that we just did not capture in these initial models.
We've had another question, and that is, did any children with late onset epilepsy have a gain of function, or they all lost of function? So in our autism families, there was a single individual who we knew had a late onset epilepsy, and that person had a loss of function mutation. To the best of my knowledge, no other case with a late onset seizure has been tested. I'm fairly sure that's right. So I think it is likely they're going to be loss of function, but this is just a model, and that's exactly the sort of thing which we need to be testing. Can a variant start as benign infantile seizures and then convert to ASD with developmental delay? So that's an interesting idea. If our model is exactly true, the answer would be no. But biology is never as simple as a model. I'm surprised that the people with benign infantile seizures seem to be so unaffected. It is extremely unusual to see someone with such a severe symptom early in life with no problems later on. It is an interesting question. If you went and really looked hard at those people, by the age of 20, when you had time for any symptoms to develop, and really did some very thorough testing, would they truly be developmentally normal? I do not know, but it'd be an interesting thing to go and do. Would I expect them to have autism? I'd expect them more likely to have, I guess, delayed developmental delay consistent with a seizure phenotype. It is possible to have autism. It'd be very interesting to know, but simply at this stage we would say we've not seen any examples of that, but we are looking out. We have to remember though that autism is a common disorder, so even if you have benign infantile seizures, there's still a 1% chance of you developing autism for other reasons. Can you speak to how de novo mutations happen? Is it when the egg and Is it in the egg and/or sperm, or is it when the egg is fertilized? De novo mutations can happen at any one of those points. The most common time for them to happen is in the egg and the sperm before conception. It is slightly more common in the sperm than the egg for the simple reason that men have longer for those mutations to accumulate in the sperm, whereas women make all of their eggs before birth, which is probably the more sensible strategy. They are basically random events, and it's one of these kind of catch-22 things. Without de novo mutations occurring, we wouldn't have been able to evolve into humans. But unfortunately, de novo mutations are sort of the rust based after that, or the necessity of evolution. We don't think that SCN2A is more mutable than other regions. We think it's just more distinctive when you have a mutation there. How can SCN2A autism phenotype lead to a severe intractable seizures late onset? Well, the simple answer to that is that in about 20% of people with autism, they do get seizures. And so it's quite common with someone with autism to have seizures. A section of those are severe and intractable. It's not that infrequent within autism cases. I suppose the deeper question, which I think you're hinting at there is the SCN2A loss of function variant more likely to lead to severe intractable seizures than someone with a different reason for autism? That is a very good question. That's something we need to dig into. It is a particularly hard one to dig into, though, because you're more likely to get a genetic test with seizures than you are without, and therefore there's going to be a big bias in the people who we can ask. We have a couple of ideas of how to do that, but it is not an easy thing to dig into. Can a variant with BIFs convert to ASD? Would they still be gain of function? So basically what we're asking is, can a variant change from loss of function to gain of function, or the other way around? Anything is possible. We've only characterized 20. It is a model. We expect there to be exceptions, and those exceptions will tell us something. We've already seen some suggestion that a mutation hitting the ion selectivity filter could have both gain of function and loss of function effects. There may be others like that, and that's something we need to look into more.
Are there treatments to reduce action potential firing? Are there any to increase action potential firing? Well, certainly there are epilepsy drugs that act directly on sodium channels or indirectly on other channels to reduce the generation of action potentials within the brain. Those that increase action potential firing, that hasn't been explored in as much depth, largely because we've just started to realize the role of sodium channels, loss of function in sodium channels in these various disorders. Also, it's a much trickier problem. You could imagine that for a given dose, you have to be very, very careful with how much you give a child. If you give them too much, you might actually induce an epileptic event, a seizure event. And so this is something that many, many smart people are interested in, but I don't think we're there yet.
I think that relates. Excuse me. That relates to this question about, is it appropriate to use supplements? You give an example here of amino acids. I wouldn't expect individual amino acids or supplements to have any effect. You're not going to be able to change the structure of this protein based on dietary supplements. There's a sort of broad question here of, can we try and find things, and I think we had some pre-submitted questions about caffeine and the beta agonists, which might have an effect. I would urge caution in looking at the biology and thinking of simple dietary modifications which might make a difference based on that biology. You're dealing here with a very, very complex system where a sodium channel is an integral part. While sometimes you can predict an effect, it's very difficult to predict the direction of effect. There could be an inhibitory factor, a regulatory factor, a feedback effect, which means that what makes sense on paper doesn't make sense when you actually do it in biology. On the flip side, if something works in your child, who cares about the biology? If it works, and that's great. If several of you are finding something works, please let us know, and we will take that into account and see if we can understand why. We have a question here, are de novo mutations in all eggs or just one? De novo mutations are so rare that they basically affect, in this situation, just one egg. Unless there's the very rare thing where the person has a somatic mutation which affects a subset of their cells, in which case it might be in more than one egg. In males, it's a little bit more difficult because as you get older, you accumulate more and more mutations in your essentially sperm germline cells, and so therefore multiple sperm get affected, but it's still a very small proportion of all the sperm. From a very practical point of view, if a child has a de novo mutation, the chance of a sibling having that same de novo mutation is 2% or less. So it can happen, but it's very rare. Good. I think that's all of them. I hope those have been useful answers. And we think we're passing over to Leah and Angie.
It's actually Lindsay again, the Simons VIP, and that's all the time we have for today's webinar. Thank you so much to Dr. Sanders and Dr. Bender for an amazing presentation, and to everyone watching for joining us today. A special thanks also to Leah and Angie for moderating the presentation and all of their organizational help to make this webinar possible. You'll be able to access the recording in about a week or two on the Simons VIP YouTube channel. We'll share out the link to all of the community once it's posted. And the links and presentation are all going to be emailed to everyone at the end of this meeting. So for anyone interested in looking over the presentation again or checking out the appendices that were included, you'll have all of that emailed to you shortly. So thanks again, everyone, and have a great day.